thank ISMB and the ECCB to offer this opportunity to present this work here. And this work is about protein structures. And here we are going to refer tertiary structures represented by three-dimensional coordinates of residues and atoms. And our method could take care of three types of protein structures. Taking the example here, this shows a protein ligand complex structure. And the red part is the ligand. And we can take care of the whole protein structure, which is everything except the ligand. And we can also take care of a pocket structure, which is a concave solvent accessible, which is contacting the pocket. And also we show the second largest pocket here to demonstrate that normally it has been observed that the largest pockets tend to carry the function of the protein. And also we can take care of the interaction interfaces, which is the residues, interacting residues between the protein and the ligand. So this includes not only the pocket, but also the interacting atoms or residues from the ligand part. And it's important to learn these protein structures because if two proteins have similar structures, they tend to carry similar functions on the similar evolution history. So this writes a pairwise protein structure alignment problem. The goal is to evaluate and demonstrate the similarity between two protein structures. And typically, it can be divided into three sub-problems. So if I think about my two hands as two protein structures, first, we need to superimpose them. So we kind of translate two hands together and then rotate them so they kind of overlap. This is called the superposition. And the next step, we need to find the corresponding residue pairs, which means which fingers should be corresponding ones. And this is called the residue matching problem. And next, we need to evaluate the superposition and the residue matching by a scoring function. And unfortunately, if we do not know the superposition and we do not know the optimal residue matching, we want to solve both of these problems together. This problem becomes NP hard. This is why heuristic methods are adopted in existing state-of-the-art methods. So in this talk, I'm going to compare our method with the TM-Align family, which is one of the most widely used method in protein structure alignment. So before summarizing their method, first let's try to learn the pockets a little bit. Let's take this residue indexes as an example for a pocket. So this pocket has 5 residues. The indexes are next to each other. That means they are connected by the peptide bond. So they can be treated as a fragment. So this contains 3 fragments. The first fragment contains 3 residues, and the second, the third fragment contains only 1 residue. And we try to calculate the frequency of the number of fragments and the size of the fragments. And we can see that pockets contains a large number of fragments. And these fragments are extremely short. This is the opposite of the whole structure alignment because normally for the whole structure is a single chain, which means it's a single fragment, long single fragment. But here it's highly fragmented and the fragments are extremely short. So this writes the importance of sequence order independence. Let's take a look at these two interfaces as an example. You can see that the X, Y, Z interacting to each other are quite similar to the X prime, Y prime, Z prime interacting to each other. And if we connect the C and N terminal, we can find 2 circles which are quite similar. This is called the circle permutation. But if we disconnect them, we can see that in the first, on the left side, the X is in front of Y on the sequence. But on the right side, Y prime is in front of the X prime in the sequence order. So this is why sequence order is not, is very important because it's not always conserved in similar interfaces. And moreover, it's not clear to us how to define the sequence order if we are dealing with residues from different chains. Because, for example, for the DNA structure, they always have 2 strands. It's not clear to define which strand should be in front of the other one. So for this reason, TM-Align tried to improve the alignment by adopting a sequence order independent method algorithm. And I'm not going to discuss the details of these 2 algorithms, but there's one thing that you need to remember for this talk. The sequence order independent method is much slower than the sequence order dependent method. For this reason, TM-Align tried to address interface alignment in 3 stages. In the first stage, they perform initial superposition sampling. These superpositions are sampled by doing initial primary and secondary structure alignment, which are long sequence alignments like BLAST-P, and this has a strong dependency on the sequence order. And they also perform a fragment tertiary structure alignment, which try to minimize the RMSD between the 2 fragments. And this has a relatively weaker dependency on the sequence order because fragments are relatively shorter. And in the next stage, they try to find good superpositions from the initially sampled ones. So this is done by calculating, by first performing the residue matching and then calculating the alignment score. Because it's possible that there are millions of superimpositions sampled from the first step. So second step, if we try to calculate the alignment score for each sampled superposition, this could be quite time-consuming. And especially if we use sequence order independent residue matching, it will be even more time-consuming. This is why TM-Align didn't, and iAlign didn't try to perform sequence order independent alignment in the second stage. So they only perform that in the last stage before producing the final alignment. This is more like a sequence order dependent alignment method wrapped by a sequence order independent refinement. So the sequence order dependent part is actually TM-Align method, and after this wrapping, it becomes iAlign method. So our method is try to improve TM-Align method based on this key observation. So we found that, let's try to think about my hands again. If I have hundreds of fingers, I do not know which one of them are corresponding ones. I'm not quite sure how to superimpose them to overlap them. But if we only superimpose 3 fingers at the same time, this becomes much easier problem. And simultaneously, it's possible that we find all the remaining fingers are also overlapped. And from our observation, we find that such superimposition almost always exists, which means even in the worst case, we can find a near optimal one. And also, such superimposition can be sampled efficiently. And we call this our method PROSTA because this is a simple method built based on a simple observation, and PROSTA means simple in Polish. So this is our high-level outline of our method. And the main difference between our method and TM-Align family is how we initially sample the superpositions and how we try to find the good candidates. And so I'm going to focus on these 2 parts in this talk, and I'm not going to discuss the last step because it's quite similar to TM-Align. And in the first step, we try to remove the sequence order dependency from the initially sampling. So we didn't perform sequence or secondary structure alignments. Instead, we introduced some new types of subsets. So we just superimpose these subsets together to sample a superposition. And here we describe 2 kinds of subsets, and this is defined for the ISMB paper, but in the next paper, we are going to introduce more subsets, which are more biologically meaningful. And for this figure on the left side, we show a local consecutive subset. So it's basically one fragment from the first structure and another fragment from the second structure. And this is also used by the TM-Align method. And this kind of modeling the local pattern of the protein structures. And on the right side, we introduce a new remote consecutive subset, which is basically 2 relatively shorter fragments from each protein structure. And the advantage of such a remote consecutive subset is that they can model remote contexts and remote topologies of protein structures. So we compare the alignment scores sampled from the initially sampling step. And if we compare the highest alignment score sampled from the remote consecutive subset and that from the local consecutive subset, we can clearly see that the remote consecutive subset tend to outperform the local consecutive subset. But sometimes a local consecutive subsets are also performing better, but this is fine because we are not trying to find a single type of subset which is best. We can simply combine these subsets together in the initial sampling step to achieve the best result. So next, I'm going to describe how we perform the superposition filtering step. So the goal is to find some promising superpositions for the following steps, but we do not want to actually calculate the alignment scores in this step, because by calculating the alignment scores, it will become time-consuming. And if we perform sequence order independent residue matching in this step, the time complexity will be unacceptable, not practical. So here shows one case study. So each dot in this figure represents a superposition. So the coordinate of this dot is calculated by applying the rotation matrix of the superposition on a unit vector. So these dots relies on a both surface, and each of them represent superposition. And we can clearly see that these dots are not evenly distributed from the both surface. And it has been, we observed that the optimal superpositions tend to lie close to the high density regions. So we try to identify the high density regions by doing clustering. So for this case study, we only keep the 4 largest clusters. That means we remove 81% of the dots from this figure. So we just need to work on these 4 clusters. So that's 21% of the sampled superposition. And the optimal superposition is contained in this largest cluster, which contains 13% of the all sampled superpositions. And so we do not need to calculate the alignment scores to identify the optimal superposition. And another advantage of this approach that is we have 4 clusters, so we can try to do refinement, try to do one refinement for each cluster. So we can perform, we can produce multiple alignments, one from each cluster, and we can kind of have some requirements saying that these clusters, because they are sampled from different clusters, the superpositions are different from each other, so the final alignment tend to be different from each other. This is not provided by iAlign family. So we try to evaluate our method on this test set of 312 protein DNA interface structures. These structures are non-redundant and of high quality. And we evaluated the quality of the alignments by using ITM score. ITM score is a modified version of the TM score, and it's also introduced by the iAlign method. So we didn't try to invent new scoring functions in our method. We just try to improve the alignment method, try to find the best way to find this optimal alignment. And then we compared our alignment scores with those calculated from TM-Align and iAlign. And it's worth mentioning that for TM-Align and iAlign, they only accept protein residues as input. So we have to perform a black box pre-processing to mask the nucleotides as residues. Also, we need to mask the chain IDs, and update the sequence numbers as well. So next, we compare the alignment scores from PROSTA and TM-Align. We are not trying to show that TM-Align is not a good method. We are trying to show that by using a sequence order dependent method to perform interface or pocket alignment, the result will not be optimal. So next, we compared the alignment scores from our method to those calculated by iAlign. So both of the methods are trying to do sequence order independent alignment. And because iAlign is still using the sequence order dependent methods in the initial sampling and superposition filtering step, so they couldn't find the optimal alignment for some cases. So their speed up strategy is probably fine for highly similar alignments, for highly similar structure pairs, but for the pairs that are not so similar, they tend to miss them. Actually, there are 483 cases that one of the 3 tested methods found reported a TM score which is higher than 0.5, And PROSTA discovered 482 of them. That means we only miss one of them. But iAlign missed more than 100 of them. So this is significant improvement. Next, I'm going to show 3 case studies. So first, I'm showing the blue structure is a ROX protein, and the red structure is a heme protein. The blue structure contains a winged helix motif, and the red structure contains a helix turn helix motif. And we can see that we found some similarities between these 2 interfaces. And the loop regions and DNA strands tend to overlap very well. And this is supported by the alignment score and the number of conserved contacts. If we check the IS score, we reported a score of 0.42, which is higher than 0.4. 0.4 is a common threshold used to distinguish similar and dissimilar interfaces. And this is significantly higher than the one found by iAlign. And also, we found 92 shared contacts between these 2 complexes, but iAlign can only find 31 of them. Another thing to note is that this blue structure has 2 interfaces, one on the top, one on the bottom. See, the top one is A, the bottom one is B. We align the the interface to A, but iAlign tend to align the interface to the one below, which is B. So this is this is showing the second advantage of using clustering. We can do refinement on both interfaces, but they will always pick one interface to do refinement. But because the initial sampling is not always optimal, so picking a single case, a single superposition for refinement is kind of risky. So this is why we could discover the correct one. And also this result is also supported by literature, because both of these proteins are playing important roles in selective DNA recognition. And next, I'm going to show another case showing that, again, we found we have a higher alignment score compared to that of iAlign, and the number of conserved contacts is higher comparing our alignments and their alignments. Also, this is supported by literature because both of these proteins are playing roles in DNA repair. And in the third case, we demonstrate a Protein-RNA mimic demonstration. And on the left side, we see the elongation factor P protein and the PROX-A complex. So the elongation P is the RNA strand here. And on the right side, we can see the second structure, which is the tRNA as per RS complex. And we have So this is the elongation. On the right side is the elongation P factor, P PROX-A complex. On the left side is the tRNA as per RS complex. So we recent work reveals the ministry of EFP on the tRNA structure. So we try to compare these interfaces by using our method. We can see that we find the correct interface, and the RNA strand and the loop region of the protein structure are overlapping. So this suggests that we could find this some similarity between these 2 interfaces. But if we use iAlign, we can see that on the left side, this structure has 2 interfaces. Again, they fail to find the correct interface. They tend to align the interface from the right side to the one on the top, which is not so similar. So in conclusion, we presented a new work to perform sequence order independent alignments between protein interfaces. And we show that we have to specifically design a method for this kind of problem. And our method has three main advantages because we employ not only the local structure signatures, we also employ the remote fragment pairs to model remote structure context and topology information in the initial sampling. And we use filtering to, we use clustering to filter the promising superpositions in our method. And we generate multiple non-redundant alignments by doing clustering as well. And I would like to thank you for your attention. And if you are interested, you can try our web server, and you can also check our group website. If you are interested, you can also apply a job in our group. And that's all. Thank you.
We have time for one quick question. Have you tried the software Click, which was developed to superpose interfaces?
Oh, sorry, what kind of Click?
The software Click, developed by Madhusudan in Singapore. You haven't tried that?
Not yet.
Okay.
Yeah, we can try compare our method with that one.
Okay, that's topology independent, and it may be nice to try it and compare against yours.
Yes.
And how fast is your method? Can be applied in a large scale?
I think our methods, the runtime of our method is comparable to that of DALI and iAlign. But again, SSM and TM-Align are still the fastest method.
Okay. So we thank all speakers of this morning's